Okay, so how can you tell if someone really is synesthetic? How do you know they're not just being artistic or metaphorical? So one of the things we did in my lab is we launched a website called synesthete.org, and it's a full battery of tests that are freely available. Please, if you think you're synesthetic, go to the site, take it. What we can do this way is rigorously find out exactly who's synesthetic and who's not. And here's the idea. It all has to do with internal consistency. So what we do, for example, is we show you a letter of the alphabet, and you have this color palette here. So by moving this slider, you can get any hue, and by moving this slider, you can get any saturation of that hue. And what you do is you pick the color. You find out of 16 million different colors that you can capture here, you find exactly the one that best matches your synesthetic percept for that letter. And when you found it, you click submit, and then you get the next letter number. You get, let's say, the number 3 or the letter X or something, and you find exactly that color. So the trick is we present you with each letter and number 3 times each in random order. And so the idea is if you're really a synesthete, you're going to find exactly that shade of color for, let's say, the letter T every time. And if you're not a synesthete, it turns out it's very hard to fake that. So here are the results for an example synesthete and a control subject who is asked to fake synesthesia. So what I'm doing is I'm plotting the colors that were chosen on each of the 3 trials, and the bar off to the side here represents the amount of variance in color space. In other words, the amount of wobble in the colors that they chose. And what you can see is that for a real synesthete, it's easy every time she's presented with the letter T, she picks the same color, or E, the same color, D, and so on. Whereas for somebody who's asked to use free association or memory, it's really difficult. It's a very hard test to pass, to remember what you said a whole bunch of trials ago and find that color again out of 16 million possible. So this is one way that we distinguish who's really a synesthete and who's not. We actually have several levels of testing to make it absolutely uncheatable. So for example, after we've collected your colors, then we flash at you on the screen, let's say the letter H. We flash an H that's either orange like this or a different color from somewhere else in your palette. And you have to say as quickly as you can whether it matched or didn't match. So for a real synesthete, if you flash, for this subject, if I flash an orange H, they'll say, yeah, that matches. And if I flash a green H, they'll say, no, that doesn't match. But for someone who's just faked their way through something, they're terrible at that, both in terms of accuracy and reaction time. And so this is just a second level of how we can very sensitively find out who's synesthetic and who's not. We've now translated the battery into many different languages, including Chinese and Hebrew, as well as a whole bunch of European languages. And this way we can find out what's going on not only in French and Spanish and German, but also in different alphabetic systems, and I'll touch on that a little bit. So what we have at this point is the ability to test all sorts of different forms of synesthesia, and what we have now is about 18 different sorts of tests, and with this we can really start getting numbers on this, because it turns out that essentially all of the literature up until now on synesthesia has had a sample size of 1. I mean, up until the early 2000s, it was all case studies, single case studies. And then people started publishing papers with an N of 2 or 4, a couple of them have 8 people in it. And what we've done now is kind of change the game on that because we've rigorously verified over 6,000 synesthetes, and we've got their data in exquisite detail. And so this allows us to sort of change the playing field a little bit. And so what I'm going to do very briefly, because I know this is a mixed audience, I don't want to kill you guys with graphs and charts and stuff. So I'm just going to give a surf over the top of some of what we're doing with this and how we actually make progress here. So I'll just show you a few examples. One of the things that started interesting me is when you look at colored alphabets, what you find sometimes is that there are certain letters that aren't colored. So this person will say, oh yeah, my F is light blue, my G is whatever that is, dark gray, my H is orange, I doesn't have a color. J is purple, K, L doesn't have a color. Like it's just obvious to them that some do and some don't. And I started wondering about that, what's going on there? So I started looking at that in more detail, and here's what I did. I took all of the letters that people either said had no color or they called them white, gray, or black. And I just rank ordered how common that was. And so if I just lump all these together and call them uncolored, what I'm showing here is the rank ordering of these and how many observations that there were. And what you see is that certain letters are appearing over here, and I wanted to understand why some letters were more likely to be uncolored, and some letters were very likely to be colored. So I'm just going to take this and flip it around here. So this is now the likelihood that they'll be colored, right? So these guys are really likely to have colors, and these guys aren't. So what I wanted to do is understand whether that had something to do with the order in the alphabet, or whether it had something to do with the frequency that these letters appear in the English alphabet with. So E, T, A, these are the most common appearances all the way down to the least likely. Long story short, it turns out that it sort of correlates with both, and that's because these two are correlated with each other. And if I remove I and O, which are real outliers here, it turns out that these are now both highly statistically significant here in terms of their correlation. So there's something going on with the order in the alphabet and or the frequency in the English language. But I started wondering about the I and O and what's going on there. So what I also did is I turned to numbers, and I looked at what people submitted as no color or white or gray or black with numbers. And what you can see when you lump them together is that there's something really special going on with 0 and 1, where those tend to be uncolored. This is just a side note, but it turns out that you've got a really high probability of these guys being white and these guys being black here. But nonetheless, these tend to almost always be uncolored, either white or uncolored. And in fact, for those of you who care about this detail, what I'm doing here is what's called multidimensional scaling, where I'm projecting this giant color space onto 2 dimensions, and all I'm asking is, how close are these guys in color space? So D and 8 come out pretty closely colored to each other. This is averaged over 1,600 synesthetes here. These are all sort of close in color to each other, but it turns out that I and 1 and O and 0 are always way off in their own neighborhood. So whether we're talking about the hue of the color or the lightness of it, how bright it is, it turns out that 0 and O and I and 1 are always way outlying. They're totally different than the others. So why is that? Well, I started wondering about that question, and I came up with 2 hypotheses. One of them is that maybe there's something special about 0 and 1 beginning the number sequence. Maybe that has something to do with why they're uncolored. There's another possibility, which is that they're both natural shapes that you experience when you're growing up, right? So well before you learn the alphabet or the number system, you're seeing lots of circles and you're seeing lots of lines. Okay, so it could be because they're natural shapes. And so the question that I then had to confront is, how would I study this? So I decided what I would do is look at a different number system. So I turned to Chinese numerals, and what you're seeing here is 1 through 10, and these are just 2 Chinese synesthetes that I'm showing for example. And there's an interesting trend here. This is still preliminary data, but what you can see is 1 doesn't have a color, and neither does 10. It ends up being gray here, and these end up being white here. Those are achromatic, consistent with the idea that these are natural shapes that you might see growing up. The question is, what about 0? Well, in Chinese numerals, that's the 0, and it turns out that that's colored. So this, at least in a preliminary fashion, supports the idea that it might have to do with the natural shapes. So I just wanted to mention that as an example of the kinds of things we can look at when we have a lot of data on this. I also wanted to mention something with timbre, instrument timbre and color. So one thing we started getting interested in was, what is it about the timbre of an instrument that maps onto, let's say, its lightness, how light the color is? So what we did is we got these recordings from the Dallas Symphony Orchestra of Twinkle, Twinkle, Little Star, played on a whole bunch of different instruments. And so this is what we played for synesthetes who are sensitive to timbre color. And we asked the question whether the lightness of the color that they synesthetically associate has to do with the high frequency content in the sound. And it turns out that's exactly what's going on. So if we measure the high frequency energy in the different instruments, you find that things like triangles and cymbals and glockenspiel and so on, the higher the high frequency energy in it, the lighter the color that gets associated with it. So a synesthete might call these, white or silver or some sort of, yellow. And down here, when you're talking about tuba and bass and clarinet and piano, it turns out that those are much darker colors that they're associating with it. Now, this is an average of 69 synesthetes. It turns out that these sorts of correlations, you find some people have it very strongly, and some people don't really have it at all. When we average over a lot of people, what we're plotting here is the correlation coefficient, which means how slopey that line is there. What we find is that most people have that positive slope, and the red ones are those that are statistically significant. So we find that on average, this is a real trend that something about the high energy frequency in the auditory domain is mapping onto the lightness in the visual domain. Yeah? No, we haven't tested this on controls, but that's a great question because I think it's likely to come out maybe similar in controls. They wouldn't be able to pass the consistency testing. But if we just asked them a one-shot thing, which is, hey, what color would you associate? I'll bet it would come out the same. Thank you for that question. I'll get back to that on my last slide, too. OK. So it turns out that synesthesia runs in family trees. And this was first noted in a paper in the journal Nature in 1883. And what happened was, that was actually by Sir Francis Galton, who was Darwin's cousin. He was looking at some synesthetes, and he just sort of speculated that was true. And it turns out he was right. What happened was synesthesia essentially slipped out of the scientific spotlight for about 100 years. And this was because of the behaviorist school of thought, where the idea was, look, we're input-output black box reflex machines, and you're not allowed to talk about private subjective experience. And so as a result of that view, synesthesia just totally fell out. But it came back recently, and so one thing that my lab is doing is trying to pull the gene for synesthesia. Now, of course, it might be different. It might be many different genes that are involved, but so that's what we're searching for. But something struck us at the beginning, which is, there are so many different subtypes of synesthesia. Is this actually the same thing? I mean, are all synesthesias due to maybe a single gene, or are they different types? Are there different types of genes involved? So here's how I went about studying this. What I did is we tested almost 6,000 people here, and we can test them on lots of different things and asked a very basic question, which is, how do these things cluster with one another? So if you've got colored letters, what's the likelihood that you've got instrument timbre synesthesia, or colored months, or colored notes of the scale, or something like that? How do these things clump with each other? Well, right away we started seeing some very clear patterns. So here's just an example. If you have colored graphemes, which is letters and numbers, then you're really likely to have colored weekdays and really likely to have colored months, but you're not really terribly likely to have any of these other forms of synesthesia. You're not really more likely than anyone else in the population to have those. So we did this sort of analysis on all the different types of synesthesia, and I'm going to skip all the math of it. We did something called common factor analysis. What we find, though, is a very simple result to explain, which is we find that it falls out into 5 clusters. So it turns out if you have colored letters, you're really likely, about 80% likely to have colored numbers or weekdays or months, any of these. If you have any one, you're likely to have another. But you're really not terribly likely to have pain to color. And if you have touch to color, you're not really likely to have sound to touch or any of these. They fall out into very different groups. And so this is really important because what we now think is that synesthesia may not be a single thing. It may be at least five different things that we're all putting under the same umbrella. And the reason we can put them under the same umbrella is because they have this thing in common where there's a mixture of the senses, but they might actually be underpinned by different genetic bases. So the one that I've been concentrating on is this one, which I've now given a new name to, which is colored sequence synesthesia. Because the really important thing to note here is that these are all overlearned sequences. That's what letters and numbers and weekdays have in common, is that they're things you learn when you're about 4 or 5 years old, and they're overlearned, and they're particular sequences that have an ordinality to them. And these things all seem to trigger color. So what we've been doing then is looking at the genetics just of colored sequence synesthesia. That's how we're deciding who we're including in the family tree and who we're not. We're taking those people who have colored sequence synesthesia, and it turns out you can look at the way this marches through a family tree, and then what we do is we take the DNA from everybody and we do what's called a family linkage analysis, which is where you look at how the small genetic changes march through the family tree, and you find out which ones are tracking with the synesthesia. And in doing that, what we've been able to do now is pull out a hotspot on chromosome 16. It's about 23 megabases long. And what we know is that the gene exists somewhere in there. So there's about 400 genes in this region. What we're doing now is narrowing it down. But we're finally getting close. And we're maybe 6 months or a year away from knowing what the actual gene is that's involved. There are essentially 2 hypotheses about what's going on in synesthesia. The one that's been on the plate for a long time is this idea that maybe what's going on in synesthesia is you have increased wiring in a synesthete's brain. So let's say the areas that care about letters and the areas that care about colors, since they're next to each other, you have more physical wiring there. That's the first hypothesis. I actually favor a different one, which is that the wiring is the same in everybody, and what's going on in a synesthete is a slight imbalance of the inhibition and excitation, such that activity in one area then kindles activity in the neighboring area because there's not quite enough inhibition. And the reason I favor this is because what I mentioned before, non-synesthetes can have synesthesia with drugs. People can have synesthesia sometimes. If you're a non-synesthete, you can have it if you're very fatigued and somebody slams the door, you can see colors. And also for synesthetes, their synesthesia waxes and wanes a bit. So when they're stressed or fatigued or on cigarettes or alcohol or antidepressants or antiepileptics, all these things will change the quality of their synesthesia. And of course, that has the feeling of something to do with the chemical balance, something not to do with the hard wiring. So I think when we find the gene, we'll be able to show that it's something associated with inhibitory neurotransmitter receptors, and we'll finally be able to put that to rest. What I'm really excited about with this is what I'm calling this pursuit is perceptual genomics, and this will be the first hit in that. And the idea is everybody around the world is pulling genes for eye color and height and weight and aortic stenosis and diabetes and things like that. But what hasn't been done is pulling the genes for how you see reality, how you see the world. And what's amazing about synesthesia is that what might be a single nucleotide change changes the way that you see the world. It changes the way you have your reality. And so this will be the first hit where we're able to say, look, you change this little tweak here, and now you're seeing the world differently. I just want to give a flavor for those of you who are in neuroscience, or for those of you who aren't in neuroscience, it might be an interesting flavor also, of how we go about looking at the neural basis of this with neuroimaging. So the question I wanted to ask once we discovered that it's all about these overlearned sequences is, well, is there something in the brain where there's something special about that? Because it's not only colors that get triggered, but I mentioned overlearned sequences can trigger these spatial forms where you have February, March, April, and so on, and they have personification where they'll trigger personalities and genders, things like this. So there seems to be something very special about overlearned ordinal sequences. So what we did is we used neuroimaging to look for that. So we had people in the scanner compare ordinal sequences like months and weekdays and letters and numbers to other things that are categories but have no order to them, like fruits and animals and car makes and furniture. I'm not going to go into any detail on this, but what we find is an area in the right side of the brain, in the temporal lobe, that really cares about sequences as opposed to non-sequences, things that have ordinality to them. So just as an example, if we're looking at the back of the brain here now, so you're looking at the back of somebody's head, if we're presenting non-sequences like animals and fruit and cars, what you find in the left hemisphere is that these language areas really care about the animals and the fruits and the cars and so on, but it doesn't really care very much about weekdays and months and so on. And what we've discovered is this right-sided area that really cares about weekdays and months and letters and numbers, but it doesn't really care so much about these other categories. So we were surprised to find this because usually anything that we think about with language is always found in the left hemisphere. This is for right-handers. All our subjects are right-handed. But what we found is this area where we didn't expect it at all, where just for things that are sequential, the brain in this area really cares about that. So we're just sort of colloquially calling this a sequence area, but it immediately suggests some interesting things. One thing is that sometimes you find patients who have left hemispheric damage and they lose their ability to speak and understand language. They get what's called anaphasia. And something that has never been explained is often people can have these aphasias and yet they can retain the ability to do weekdays and months and letters and numbers. That stays intact even though they've lost their ability to do the rest of language. It also suggests, the fact that there's this sort of special area here suggests a basis for why these overlearned sequences keep coming up in synesthesias, why they're so common. It seems that there's a special network that is somehow devoted to these. And so when we're asking about where in the brain do you find synesthesia, what's always been suggested in the literature up till now is that it's a left-sided thing because it has to do somehow with language. And what we're suggesting is exactly the opposite, which is that the sequence area happens to be right next to color areas. It happens to be right next to even some newly discovered color areas in the brain that are right next to this middle temporal gyrus here. And so we think it's this neighborhood relationship on the right side that matters. And this has recently been verified, or I should say this idea is supported by the fact that people, when they look at the amount of connectivity, when they look at the fibers in the brain, they find in the same area that we do, on the right side, they find increased connectivity in the synesthetic brain. And so the suggestion now that we're making is that synesthesia is actually a right hemisphere phenomenon that has to do with the sequence area. And so the last thing I'll mention is, coming back to these spatial sequence synesthesias, again, this is overlearned sequences. You've got months and weekdays and numbers and so on. You've got these having spatial locations. One thing that we've done in the lab is set up virtual reality software where we can drop you in as an avatar, and you can arrange the weekdays and the months and so on according to how you actually see them. So these, for those of you who can see, these are the years, 1945 and 50 and so on. You can arrange the years or the number line or the months or so on exactly how you're experiencing them. We also have an online version of this. If anyone wants to go on the site and do this, you can take a third-person perspective or the first-person perspective, and we've collected a whole bunch of data on this now. Yes, that's a great question. They always are. So there's something about being neighbors in a sequence, like January, February, March, that maps onto neighbors in space. And it may be that somehow your brain is doing this as a way of learning the sequence. Yeah, it's a good observation. So what has been in the literature about spatial sequence synesthesia, especially as regards weekdays and months of the year, people have called this time-space synesthesia. And there was a paper that had as the title, Ovals of Time, as though this has something to do with time. And that's just a really wrong-headed way to think about it, because in fact, we don't just find it with weekdays and months, but you find it with letters and shoe sizes and the Indian caste system and temperatures and primetime television lineups on Thursday night and so on. Essentially, it's any overlearned sequence is what triggers this. It's all about overlearned sequences. So this whole idea of tying together time and space is just really wrong-headed, and we don't ever have to talk about it again, because that's what it's about. And so what this gives us is a new anatomical view here, where this on the right side of the brain is the part that cares about overlearned sequences. That happens to be right next to the parts of your brain that care about objects. It cares about objecthood. And what appears to be happening in spatial sequence synesthesia is that these ordinal sequences get tied right into this notion of objecthood, and so that's what's going on. It's like it becomes a reified thing. It becomes an actual object for the person. I'm going to switch gears for just a minute and tell you about two shapes that were just found in a South American tribe. So they found this tribe that had no written language, but they do have these shapes, and one of these shapes is called Kiki, and one of them is called Buba. And the question is, which one is which? Who thinks that that's called Buba? And who thinks it's called Kiki? Okay, and that's called Buba. Okay, great. So it turns out I lied. There is no South American tribe. These are just two shapes. This was introduced by a psychologist in the 60s, and more recently Ramachandran has been showing this. The idea here, the really important thing, is that what I've shown you is visual shapes, and there's a natural mapping onto the auditory domain, where you think, oh yeah, it's totally natural. That should be called Buba, and that one called Kiki. Well, that's like a synesthesia, right? I mean, it's a weird cross-modal thing that everybody does. So there's this open question that we've been dealing with, and in fact, in my book, which I'll mention in a second, this is how we culminate the book, is this question of, does everyone have synesthesia in some way? So as an example, if I were to go to a piano and hit the high note, ding, 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 and low note, dong, 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 and I ask you, which one is brighter? Which would you say? Okay, and if I were to ask you, which one is bigger? What would you say? Okay, well, everybody answers that, and that's so strange, right? Because all I'm doing is playing some notes, and you're mapping that auditory signal onto brightness and onto size, right? So it's as though the connectivity between these areas might exist in everybody, this sort of cross-modal domains. And in fact, when you look at the microanatomy, what you find is that there are fibers carrying visual information that go into the auditory part of your brains, and there's fibers carrying auditory information that plug straight into the visual parts of your brain. This is in everybody's brain. So it turns out there's a lot of mixing of the senses anyway, and I think that's why we get certain synesthetic things. For example, there are many expressions in the language that were presumably introduced by synesthetes, like a loud tie, or a cool jazz, or a sweet personality, or a loud, I already said loud tie. Anyway, sharp cheese, there's another one. There are all sorts of these expressions that really represent some form of synesthesia, and it kind of works with everybody. We all kind of know what that means when you talk about a loud tie. So this is the sense in which we all might have synesthesia, but we all might have this cross-modal connectivity, but only about 1% of the population is actually able to access that consciously. Okay, I'm just going to summarize here, and then I'll take questions. So I started off by showing you that reality is really a construction of the brain. And synesthesia is a really good inroad into us understanding how brains construct this. Because if we're just trying to understand our own reality, we're like fish in water, right? It's really hard to describe the water because we've never seen anything other than that. But when you see a different way that it can be, that gives you a lot of traction. I mentioned that many artists have leveraged their synesthesia. Synesthetic brains have more crosstalk, where neighboring areas are talking to each other more. And one thing to go home, if you forget everything else, is that not everyone sees the world the way that you do. I'll just mention, I just published a book on this with my co-author, Richard Saitowick. It's called Wednesday is Indigo Blue. And this is everything that we know about synesthesia. And this is written for a general audience, and it's full of stories, and it's printed in full color. The other thing is, if you think you're synesthetic or you know somebody who is, then send them to synesthete.org. Thanks very much. All right.